Books on Tape presents DNA, The Secret of Life by James D. Watson with Andrew Barry. Read by Dan Cashman. Introduction. The Secret of Life. As was normal for a Saturday morning, I got to work at Cambridge University's Cavendish Laboratory earlier than Francis Crick, on February 28, 1953. I had good reason for being up early. I knew that we were close, though I had no idea just how close, to figuring out the structure of a then little-known molecule called deoxyribonucleic acid, DNA. This was not any old molecule. DNA, as Crick and I appreciated, holds the very key to the nature of living things. It stores the hereditary information that is passed on from one generation to the next, and it orchestrates the incredibly complex world of the cell. Figuring out its 3D structure, the molecule's architecture, would, we hoped, provide a glimpse of what Crick referred to only half-jokingly as the secret of life. We already knew that DNA molecules consist of multiple copies of a single basic unit, the nucleotide, which comes in 4 forms: adenine, A, thymine, T, guanine, G, and cytosine, C. I had spent the previous afternoon making cardboard cutouts of these various components, and now, undisturbed, on a quiet Saturday morning, I could shuffle around the pieces of the 3D jigsaw puzzle. How did they all fit together? Soon I realized that a simple pairing scheme worked exquisitely well. A fitted neatly with T, and G with C. Was this it? Did the molecule consist of 2 chains linked together by AT and GC pairs? It was so simple, so elegant, that it almost had to be right. But I had made mistakes in the past, and before I could get too excited, my pairing scheme would have to survive the scrutiny of Crick's critical eye. It was an anxious wait. But I need not have worried. Crick realized straight away that my pairing idea implied a double helix structure, with the 2 molecular chains running in opposite directions. Everything known about DNA and its properties, the facts we had been wrestling with as we tried to solve the problem, made sense in light of those gentle complementary twists. Most important, the way the molecule was organized immediately suggested solutions to 2 of biology's oldest mysteries: how hereditary information is stored and how it is replicated. Despite this, Crick's brag in the Eagle, the pub where we habitually ate lunch, that we had indeed discovered that secret of life, struck me as somewhat immodest, especially in England, where understatement is a way of life. Crick, however, was right. Our discovery put an end to a debate as old as the human species. Does life have some magical, mystical essence? Or is it like any chemical reaction carried out in a science class, the product of normal physical and chemical processes? Is there something divine at the heart of a cell that brings it to life? The double helix answered that question with a definitive no. Charles Darwin's theory of evolution, which showed how all of life is interrelated, was a major advance in our understanding of the world in materialistic physico-chemical terms. The breakthroughs of biologists Theodor Schwann and Louis Pasteur during the second half of the 19th century were also an important step forward. Rotting meat did not spontaneously yield maggots. Rather, familiar biological agents and processes were responsible. In this case, egg-laying flies. The idea of spontaneous generation had been discredited. Despite these advances, various forms of vitalism, the belief that physical-chemical processes cannot explain life and its processes, lingered on. Many biologists, reluctant to accept natural selection as the sole determinant of the fate of evolutionary lineages, invoked a poorly defined overseeing spiritual force to account for adaptation. Physicists, accustomed to dealing with the simple pared-down world, a few particles, a few forces, found the messy complexity of biology bewildering. Maybe, they suggested, the processes at the heart of the cell, the ones governing the basics of life, go beyond the familiar laws of physics and chemistry. That is why the double helix was so important. It brought the Enlightenment's revolution in materialistic thinking into the cell. The intellectual journey that had begun with Copernicus displacing humans from the center of the universe and continued with Darwin's insistence that humans are merely modified monkeys had finally focused in on the very essence of life. And there was nothing special about it. The double helix is an elegant structure, but its message is downright prosaic. Life is simply a matter of chemistry. Well, Crick and I were quick to grasp the intellectual significance of our discovery. But there was no way we could have foreseen the explosive impact of the double helix on science and society. Contained in the molecule's graceful curves was the key to molecular biology, a new science whose progress over the subsequent 50 years has been astounding. Not only has it yielded a stunning array of insights into fundamental biological processes, but it is now having an ever more profound impact on medicine, on agriculture, and on the law. DNA is no longer a matter of interest only to white-coated scientists in obscure university laboratories. It affects us all. By the mid-60s, we had worked out the basic mechanics of the cell, and we knew how, via the genetic code, the 4-letter alphabet of DNA sequence, is translated into the 20-letter alphabet of the proteins. The next explosive spurt in the new science's growth came in the 1970s with the introduction of techniques for manipulating DNA and reading its sequence of base pairs. We were no longer condemned to watch nature from the sidelines, but could actually tinker with the DNA of living organisms. And we could actually read life's basic script. Extraordinary new scientific vistas opened up. We would at last come to grips with genetic diseases, from cystic fibrosis to cancer. We would revolutionize criminal justice through genetic fingerprinting methods. We would profoundly revise ideas about human origins, about who we are and where we came from, by using DNA-based approaches to prehistory. And we would improve agriculturally important species with an effectiveness we had previously only dreamed of.